situation like what we have right now. So with that in mind, um, if things are, most of the diseases, especially the infectious diseases, are zoonotic in nature, it is important to basically understand the disease dynamics from a holistic approach. And that's where One Health comes into play. So in Nepal, back in 2010-11, the concept of One Health wasn't that much established. And with the initiation of our project, Predict, we realized that in order to implement our project, we really need to establish One Health. And so started our effort in putting together a One Health concept to be ingrained into policies and programs. That's the start, the beginning of the One Health initiative, and eventually the government was able to put policies in place for One Health, and now it is a part of a holistic healthcare system. So, so what is important then, that if we if we want to understand our disease dynamics, zoonotic diseases, we really need to understand what are the causative agents. So Predict looked at all the diseases that cause pandemics or large epidemics. And the conclusion was that viruses play a very important role in causing pandemics. Viruses, specifically RNA viruses, because they have the mutation, or rapid mutation capabilities or potential. And because of that, we wanted to focus on RNA viruses. Unfortunately, what we know about RNA viruses is very, very limited. As a matter of fact, with the billions and billions of viruses that are floating around, especially the RNA viruses, we know a few percentage of that. And so the main focus initially was to discover these RNA viruses in animals, in humans. Animals included wildlife as well as domesticated animals, humans, and also in environment. And hence the concept of One Health being the premise of what we wanted to do. So in case, now we've been seeing that currently with the corona part, especially with the asymptomatic portion of it, it's very difficult to detect the pathogen. Only after the clinical symptoms show up that we'll be able to then test for the pathogen. So that has been a huge challenge for the current COVID-19 or SARS-2. Previous SARS was a little different because usually after symptoms appeared, that the viruses were transmitted. And therefore, if you could contain people with symptoms, then you would more or less contain the transmission. SARS-2, COVID-19, has been a challenge because there are so many asymptomatic cases, right? So depending on how large of a scale pandemics are or the epidemics are, it's always the same phenomenon, right? So you will have exposure without infection. You have subclinical diseases, and then you have clinical diseases. Most of the detection happens in the clinical phase and shedding part that I mentioned. So there are two, in terms of viral discovery, we employed two different strategies, right? So the first one is direct virus detection and characterization using molecular methods. The second part is to look into the history of it, almost figuring out the details of epidemiology, there is this serological tools that we use to uncover the exposure or the history of the infection. Now, of course, um, not viruses are the same. Not all viruses are the same, and not all viruses becomes pandemic. So there are different factors at play um, for a virus to cause a pandemic scale disease. In the case of corona, for example, if you have human to human transmission, of course, then that becomes a strong candidate to bring pandemic diseases. So it is important then that we do an early detection of these potential viruses that could become the next SARS, next Ebola, next Corona. And so our effort has been based on the idea that we want to discover these viruses before they cause pandemics, before they cause large epidemics. Now, in order to do that, we will have to have a very holistic approach to it. And because a lot of the wild animals could be a potential reservoir for a lot of these novel viruses, it becomes important that we understand that aspect of it as well. So while we are doing the front end part of viral discovery, we basically focus on reservoirs that might have the potential to carry all these pandemic potential viruses. And so that's what we look for. And based on that, we put together our plan to sample and look for viruses. Now, it is important, though, that you want to understand disease dynamics, and that would involve first the reservoir part, second the potential to go from species to species part, third look into the potential of them doing human to human. Now, in order to do that, in order to understand the whole dynamics, take One Health approach and you do concurrent sampling on potential high-risk population. And that's what we've been doing. And it looks like coronaviruses are, a family of coronaviruses are the most diverse and most potential from a pandemic angle. So the idea, again, is to have an early warning system. That was the main goal of our project. And so in Nepal, so that's the background of our overall global project. So in Nepal, we focused on doing viral discovery in a couple of different areas. I'll talk a bit about community-based surveillance for emerging diseases. And I will also talk about hospital-based surveillance. Now, in order to do that, back in 2010, we didn't have the full capacity to do viral detection and characterization, including a full-scale RNA detection and subsequent sequencing. So we had to do a lot of capacity building in the early part of our project. And like I mentioned before, two surveillance systems that we put together. One is two types. One is community-based surveillance, emerging disease surveillance. And another one is hospital-based disease emerging surveillance system. And then we targeted two different areas. One is urban setting, and the second one is rural setting with bushmeat potential viral. So in the urban setting, Kathmandu has about 5 million people. It's densely populated. It's rapidly growing. It's one of the fastest growing cities in in Asia. And it also has areas where what we call temporary settlements or slum areas. And vulnerabilities in terms of poor hygiene and therefore disease spillover. And so we focused on these temporary settlements in Kathmandu, and we also focused in a hospital based in Kathmandu as well, where we might catch the emerging disease infections in incoming clinical um, cases. Down south, there is a community called Chepans, and culturally they have been catching bats, fruit bats, and eating them. And so we uh, wanted to understand viral diversity, the potential of um, viral spillover from bats to humans. And so we basically set up a community-based surveillance in there where we caught bats and sampled them in humans and also rodents and that sort of thing from One Health perspective. And we also set up another clinical-based surveillance in one of the hospitals close to that area, right next to the Nepal's largest national park called Chitwan National Park. So basically, overall, a huge effort in about 8 years. We collected and tested over 20,000 samples. It included a lot of field work in areas that are not easy to collect samples, and then a lot of lab work looking for viral families that could be that could harbor these potential um, pandemic pathogens. Um, and so these are some of the pictures. We caught a lot of rodents, um, rats and shrews and that sort of thing in the urban setting, also in the rural setting. A lot of animal production samples, and also the environmental samples as well. In the rural area, again, bat samples, rodents, and humans as well. And we really started specializing in fully bio safety prepared field team to go out and sample in any kind of situation, in any place within the country. So overall, like I mentioned before, we collected a lot of samples from One Health perspective of different species, and we did viral screening of them. In terms of viral screening, we kind of adopted what we call consensus PCR, basically looking for viral families, and then if we detect anything on it, do sequencing for characterization. So we primarily focused on phyllo or philo influenza viruses, corona, flavi, paramyxo, and rhabdo, retro, those sort of things. And so currently we're also using massive parallel next-gen sequencing technology to basically massively identify these viruses and characterize them in a high throughput scale. So in the last 9 or 10 years, we have identified or we have detected over 119 or 20 different viruses, 19 of which are very unique to us, not found anywhere else. So they are very novel viruses. We also found viruses, coronaviruses, in bats, rats, ducks, and humans, both alpha coronaviruses as well as beta coronaviruses. Now, these viruses, obviously, next thing that we're trying to do is do whole genome sequencing so that we can do comparative genomic analysis to identify or to discern between a potential a pathogen that has caused pandemics versus a potential pathogen that might cause pandemic. So in the case of coronaviruses, for example, we have, and I'll show you a bit later, we have identified coronaviruses in all these different species. We also have whole genome sequencing of the first case of human COVID-19 we detected in our lab. And so it's a lot of comparative genomic analysis to look into ACE2 receptors or envelope proteins, spike proteins, that sort of thing. And what changed? What changed between the coronaviruses that are found in rats and the one that is spreading that's causing pandemic? And I think it's an important question that hasn't been fully answered yet. Additionally, we do a lot of what we call risk assessments while we go out, we also get information about other ecological, behavioral, and biological risk factors, including perception of diseases in the communities. And so, mentioned before, so community and clinical-based samples, a lot of capacity building, a lot of training, a lot of surveillance systems in place. And then also understanding the holistic information about the behavioral part as well, and the interfaces where the spillover could happen. So One Health was an important aspect, basis of our emerging disease surveillance work under PREDICT. So I'll come to a bit about, after Corona started here in this country, what we have been doing to address Corona, because this is a, after 8 years of preparing for this kind of pandemic, the pandemic came to Nepal. And so what did we do to address COVID-19? So preparedness, capacity building, active surveillance, and testing. So we piloted this out in one of the municipalities in the city, in Kathmandu. And basically, in the earlier phase, we set up a high-risk surveillance system for COVID-19. High risk meaning people who have come from abroad, who are the residents. So we used extensive mapping, including GIS, to figure out where they are, get their surveys, get them to one of our sites for sampling, and then we tested them using PCR. So initially, when the pandemic was starting here, COVID was starting over here, the country was not prepared for dealing with the COVID cases. So we did a lot of training on PPE and all that for all the healthcare workers, and also provided them with a lot of PPE, and we're still providing them with a lot of PPE and continual training to make sure that they are handling the situation in a safe manner. And so we also taught them how to collect samples and that sort of thing. And then we gave out a lot of PPEs. I mean, that's something that the country was not prepared for, didn't have. And so we have been working with a lot of donors in and outside of the country to make sure that, we have these supplies to be given out to the hospitals. And then in about 2-3 weeks, we set up a molecular lab, a real-time PCR-based molecular lab in one of the hospitals, municipality hospitals, where we are doing a lot of screening now. And that's a picture of a mayor of that city who's observing how a real-time PCR works. And because we worked with the local government and there was a political will to do it, a lot of things came together really, really rapidly. We also put together a protocol in place to sample community-based surveillance. In a community-based surveillance system, these potential high-risk population using a very safe manner in a high-throughput way, right? So we could do about 100 samples per, let's say, 3 hours kind of deal, and then we test them. So with this and creating a database that includes the GIS portion of it and visual interface that are easy to look at, a lot of our decision makers, especially in the government, local government, and program implementers, were able to look at things in a much more simpler way so that the visualization helped them in making their right policies and programs. Now, of course, to do the molecular testing, we were the first to bring the WHO recommended molecular testing protocol. We kind of validated here in our lab, and then we supplied the reagents and training to the National Public Health Lab, and that's how they were able to start the COVID-19 testing in the country. Again, we gathered a lot of information about these residents that's coming from outside, uh, which we are using to figure out the epidemiological portion of it. So overall, though, there are a few components that we really wanted to put together to be prepared for COVID-19. That includes the hospital with all the proper components to deal with incoming COVID-19. Also, once we put all of this together, the isolation ward, the ICUs, quarantine part, and all that, um, we did the first preparedness drill um, for to make sure that all these different components are working in the hospital. And of course, the active surveillance part is extremely important. Unfortunately, rest of the country didn't have a good surveillance system in place. The cases are increasing now and all that. Now we are getting into environmental surveillance. We were able to detect SARS-2 in sewage samples recently, about a week ago. Now we're kind of doing environmental surveillance to identify wards or areas where these infections might be a hotspot. And so then you go out and do active surveillance to screen in much more detailed manner. Of course, the last component, but probably the most important component of that, of everything, is to inform, make aware, and educate the population about COVID-19 in general, but the preparedness also, because that gave them the confidence to deal with the problem. With information came the power to decide. And lastly, I'll go a bit in terms of what we have found in terms of corona. As I mentioned before, corona was in our radar for the last 3 years, simply because its potential to cause upper respiratory diseases and the rapid aerosol mediated transmission. And so we've been collecting a lot of these samples and screening them for corona. And like I mentioned before, we have found corona in bats in the city, rodents and ducks and humans from the same area. So now we're doing whole genome sequencing to do a comparative genomic analysis. And also on the other end of things, where diagnostic has been a problem or access to testing has been a problem, we have developed a mobile, portable, handheld PCR and genomic sequencing based platform that fits in 2 briefcases, and then we can take it out anywhere and do the quick detection and characterization. So we are using on the right-hand side of your screen, you'll see that little mobile phone with that little device. That is a portable real-time PCR device that runs on a battery. And then right next to that is a small Union device that can do whole genome sequencing. So with the real-time PCR, you do qualitative assessment, yes or no. And if it is yes, then we can take that. And then do the whole genome sequencing part. And that has been our effort so far. So now our next upcoming project is to set up a COVID-19 testing facility in the Everest region, a remote area in Everest region. So at the end, this has been a very concerted, collaborative kind of effort. And it involves our government agencies. It involves a lot of our international collaborators, especially our collaborator in the US, UC Davis, and also a lot of our stakeholders within the country as well. The few new things that we're doing in the upcoming days, of course, are doing a lot of environmental surveillance to identify the hotspot. Also really working on our portable device. We can detect and characterize the pathogen rapidly in areas that are not easily accessible or where the healthcare systems are not that great. And in terms of research, again, whole genome sequencing part is something that we're working on to do a comparative genomic analysis of SARS-2 with other coronaviruses. And because Nepal had, it seems like Nepal has a lot of these corona endemic exposures in the population. The other project that we are about to start with our collaboration from Spain, our Spanish collaborators, are to do antibody profiling that are associated with coronavirus infection and how that might have a cross-reactive potential to fend off SARS-2. And so community-based herd immunity dynamics, as well as potential, what they've been calling it now, the plasma treatment. So moving towards more of a vaccination part in the later stage of our effort, simply because we also research and manufacture vaccines in the country. So with that, I close my presentation. I know it's been a little bit over 20 minutes, but I'll be happy to take any questions that you have.
Okay, thank you, Dr. Dibesh. And it was a very nice presentation. We got a comprehensive idea on the challenges faced by scientists in Nepal regarding disease surveillance, evolution, findings, and other testing procedures. Now I hope Dr. Asha, our co-chair, will give a brief idea about the topics under presentation. Dr. Asha, please.
Thank you, sir. Thank you, Dr. Dibesh Karmacharya. It was a very entertaining and as well as educating session on the One Health activities that is being going on in the Kathmandu, Nepal area. And thank you, sir, for that wonderful presentation. So we will see what are the salient features of your presentation. Firstly, you said why you concentrated on RNA viruses, because they had the maximum pandemic potential. Also, you explained very well, sir, what is the difference between SARS and COVID-19 infection, and what is the difficulty in controlling or containing the spread in case of COVID-19. Then you moved on to what are the types of surveillance systems that you are implementing in Nepal region, like community-based surveillance system as well as hospital-based surveillance system. Both are very important because community will give you exactly an idea regarding what are the diseases that is being going undetected in your area, and especially the slums area as well as those community that were having a particular type of eating habit. So that was very it was a good type of work. Like you detected lots of viruses, and you said 19 novel coronaviruses were 19 novel viruses were detected because of that community surveillance. That is a huge achievement, sir. Then also you did the whole genome sequencing of the first COVID-19 patient, and you compared it with the coronavirus that you had found out before in rats, I think. So that was also a good work. And later on, you moved on to the work you have done in the pandemic scenario, like training and distribution of PPE kits and the collection of sample training. All these things were very important in the pandemic scenario, and you the hospital or the health sector was hugely strengthened by all these trainings. Then you also said that you had set up a molecular lab in a hospital where you can do the PCR testing easily. And you also made a protocol for community-based surveillance, which was the need of the hour. And then the testing kits were developed in your laboratory as per the WHO recommendations. Then a portable PCR machine, that is a very huge achievement, sir. affordable portable PCR machine was prepared. Then you mentioned the next projects, coming up projects like plasma treatment potential awareness, then community dynamic study, and also a COVID testing lab in the Everest region. All these things were very hugely needed in this pandemic scenario, and it was a very good presentation, sir. Thank you.
Thank you. Thank you so much.
Thank you, Dr. Asha, for giving a comprehensive idea about this topic. And now it is time for your questions and feedback. So, anybody who wishes to interact with Dr. Dibesh, they can contact the host, Dr. Irshad, or they can give their queries in the chat box also. Okay, thank you, Dr. Dibesh. Again, I congratulate Dr. Dibesh for giving us such a wonderful idea about this topic. And I am thankful to the organizers also for giving me this chance for chairing this wonderful session. Now over to Dr. Irshad for further procedures.
As Sathian sir said that the topic is open for discussions. All the participants can ask your question directly to the speaker by unmuting your microphone, or you can type your message in the chat box.
Sir? Hello, sir. Dr. Karmacharya, sir?
Yes, hi.
Hello, sir. I have a question. You said you did the whole genome sequencing and compared it with the coronavirus that is present in the rat. So what was the analysis result, sir?
So
how much analogous it was?
Yeah. So we are actually currently doing it. We have RDRP gene comparison, the phylogenetics of it, where we have identified, both beta as well as alpha coronaviruses. The whole genome part, we still are working on it. So we're using two different platforms for it. One is, they call it the Arctic primer based whole genome sequencing of coronaviruses, especially SARS-2. So we're collaborating with Los Alamos in the US on it. And the other one is we're using the nanopore, the small anion based nanopore technology to get the whole genome sequencing. Once we have that, then we will do whole genome sequencing analysis, but primarily focused on things like ACE2, the envelope gene, the spike protein, and N gene. Those are the stuff that we want to really kind of compare to look into the what do you call it, infectivity potential, and what are the differences between SARS-2 and the other coronaviruses that are floating around.
Okay. Thank you.
Welcome. I think there is a question on the chat box.
So any other questions or queries, you can ask it now or type in the chat box, please.
Yeah. I do see a question coming from Abhishek. Is disease surveillance in wildlife possible by analyzing the samples from domestic animals in buffer zone? I think that's the question. And absolutely possible. And one of the diseases that we are looking at, which affects big cats, might affect big cats, is canine distemper. It's a virus. And so canine distempers are found in dogs. We've found a lot of canine distempers in stray dogs. And so in both areas, right next to the national park, um, these canine distemper viruses could be transmitted in either direction, from the domesticated dogs to the wildcats, or from wildcats to the domestic dogs. We are particularly looking into this interface in one of the areas here where there's a river. There are stray dogs that have canine distemper, but they come in contact with wild foxes. And so there is this potential dynamic of the virus moving from either direction, from stray dogs to the foxes to the others, or vice versa. So that potential is there. And by doing is having a surveillance system on animal diseases close to the national parks or the interfaces between wild and let's say human settlement, I think we will be able to pick up a lot of these important pathogens.
Any more questions? Dr. Manoj S., can you ask directly? Please unmute and ask. So the question is, low amount of exposure to COVID-19 virus can get immunity.
Could you repeat that question again?
Low amount of exposure to COVID-19 virus can get immunity.
We're not sure, right? So if you look at the current infection rate, whether you were looking at India or even Nepal, we might have a situation where the community transmission has already happened. But 80% of these infections are, it looks like, asymptomatic, right? And so why are we not seeing the kind of mortality or morbidity because of the SARS-2 compared to, let's say, Spain or Portugal or the US to over here? I think it's a very interesting question that we should be looking into. And so we've been talking to our collaborator in Spain to look into their population and our population, looking into the antibody profiling between these two populations and the SARS-2 relevancy, both in terms of infectivity as well as the clinical implications and mortality eventually. And so I think the dynamics of your exposure, whether your endemic corona strains or SARS-2, has a lot to do with the mortality component to it. So would population-based disease susceptibility is something that would be of huge interest at this point.
Dr. Bhupendra, can you ask directly? The question from Dr. Bhupendra, can migratory bird transmit the virus, and if yes, then how should we protect our livestock and ourselves?
Right. Absolutely. So we did a research on role of migratory birds in bringing influenza A. And we looked into various different migratory birds, and we screened for influenza A using their fecal samples in Nepal. This is back like 4 years ago. And what we found was we found influenza A in a migratory bird called ruddy shelduck, which visits like the southern part of Nepal as their stopping point. And it has a huge Eurasia migratory path. So we identified the influenza A, which turned out to be H9N2, and the ruddy shelduck. And we went out and then we kind of started getting samples from chickens and ducks and all that. It turns out H9N2 is a very is a readily floating strains of influenza A in that area in chickens and in ducks, and of course ducks are notorious reservoirs for a lot of these influenza viruses, and so that possibility is actually there a lot, especially in the case of influenza. And so, and we actually have evidence for it, and this is something that we published in PLOS ONE. But in order to make sure that we don't have that, there can be some measures that you can take where the intermingling sites are, basically make sure that they are separate, right? That they're not there. Would be one preventive aspect of it. But a lot of these diseases or these viruses, they are going to kind of go from one species to another anyways. So I don't know how successful that would be, but there are certain measures that you can take to prevent it.
Dr. Ashok Pillai. The question is, even virus came from animals, but why do animals are not affected that much?
It's an interesting fact, right? So I don't think, like Corona or any other, we can't blame the animals for it, because, what we have been doing is, our population has been increasing, we are encroaching to their habitat. And take bats, for example, right? So bats are one of the oldest mammals on the planet. And there are many different species of bats, much more than many other species. And because they're such an old mammal, they harbor a lot of these viruses, and they actually coexist with these bats. It doesn't affect them that much. But it's the time when you start getting closer to bats. It's the time when, using bushmeat, what happened with the HIV, retroviruses, what happened with Ebola, with bats, and you look at all of these different cases, you see that humans are encroaching, land conversion, encroaching into their habitat, getting close to it, and all that kind of stuff. That's causing this problem, and this problem is going to continue more if we continue doing this the way we do things. So I think there is a philosophical thinking, especially with the COVID-19 pandemic that's happening now, is, I'm a conservation biologist, and I believe I put a lot of emphasis on our nature and our preservation of our natural, heritage and natural settings. We disturb that a bit, and then we are going to come across with all these different things that might not be very friendly to the humans. And so I think the philosophical question now is, what is development, right? Are those roads and more economic-based focused development is what we consider development, or are we looking into more of a holistic approach to living, where we can live being a part of the ecology and not, taking ecology as our resources? And so I think that's an important philosophical question that we have to ask.
Hope there are no further questions.
Sir, one, two questions also there.
Yeah, please.
From Dr. Nisha P. Kulkarni. Can we attain herd immunity against coronavirus?
Again, we don't know so many things about coronaviruses, including herd immunity. It looks like from some of the European data that herd immunity is not attainable at this point, even with 60-70% exposure. And there are some interesting case scenarios where we can compare how different countries dealt with the pandemic, how Korea dealt with it, how the US dealt with it, and how the European countries dealt with it. And one interesting case study could be a case study in Sweden, where they said, we want to attain the herd immunity. Therefore, we're not going to, but if you really look at the data, their mortality was a bit higher. And And I think they have not attained the herd immunity that they were looking for. So still, we don't know a lot about SARS-2. We don't know a lot about the immunological response to it. And we don't know what are the implications of the exposure to certain population versus the other, and whether a certain population has immunity already or attains herd immunity as they move forward. So I think these are interesting areas to be investigated.
Dr. Prashant Kumar. Hello.
Hello.
I request to all the participants, keep your microphone off position while you are not using it. The question from Prashant Kumar asks, the interdisciplinary collaboration is a key element for One Health concept. When we think about human diseases, we need to work with the clinician, epidemiologist, veterinarians, etc. It needs a great effort to collaborate with them. So, what challenges you have faced while working on it?
Yeah, so I think it's an important question. I mean, we keep on saying One Health is important. One Health keep on saying makes a lot of sense and all that. However, historically, if we look at it, we have not worked in harmony. Animal health has never worked really in close harmony with the human health, because the perception in many countries, including ours, is that human health is somewhat superior to animal health, which is not the case. We all know which is not the case, but we tend to work in silos. And One Health concept, as old the concept is, implementing One Health is very, very challenging, because you almost have to break these silos and reconnect them on a horizontal scale. And that's where the problem starts. But there are some good examples of where One Health worked really well between animal health and human health. And in Nepal, for example, even influenza surveillance, there is a parallel effort between human health and animal health to contain or prevent the avian influenza. But it's driven more by the need as by the philosophical concept that overall, in general, One Health is going to help us understand disease dynamics, help us to manage diseases, and eventually help us to prevent diseases. And although the concept is wonderful and all that, implementation is difficult, because what I call a human element plays into the picture, where some people feel superior than the others. There's ego flying around. There's all other interest that's around it. So it's a complicated issue. However, with say COVID-19, there's more emphasis on One Health and how we need to look. Because if you really look at this disease, that's pandemic, that's creating havoc in a scale that we haven't seen before, there are some outstanding questions we have not answered. We have not been able to answer. One, where is the reservoir? Are those bats? Are those pangolins that was intermediary? And how did that go from a meat market to being a pandemic, right? So what is the reservoir? How did the species to species jump happen? And how did this particular virus gotten so much potential to wreak havoc on a global scale in a scale that we have not ever seen before? We don't know a lot of these answers, including the immunity part, right? Why some countries are better than the others in terms of dealing with it, and why some population do fairly better or less affected by it than the others. A lot of these questions are still not being answered. And if you look at this, you realize that One Health, if we all work together to kind of understand this, manage it, prevent it, it's going to be good for everybody.
Sir, one more question from Rudresh Gowda B. why can't we use Western medicine and Ayurveda in combining to combat COVID-19? As Western medicine claim that it can cure the disease, and Ayurveda claim that boosting of immunity. That's interesting.
That's an interesting question. I think I'm of the opinion that I do science, and in science what we do is we uncover evidence, and only with evidence we can claim this or claim that. Without that, everything else is presumption, and presumption might work in certain percentage, but in large percentage, doesn't work. It becomes more subjective, right? And so to answer that question in a very the way I can understand that question is, I'm of course open to the whole idea of the modern medicine, what we know about and how we can do it, and also the Ayurveda, right? So a lot of the modern medicine, penicillin, for example, penicillin, comes from, everything comes from nature. So I'm open to the whole idea. We have to look at all different possible solutions.
So there is one last question. The probable whole genome sequencer you are using is really fascinating. Is it based on nanopore technology? The question from Serin Mary Pierre.
Yeah, so there are two methods we're actually using. One is the next-gen sequencing based on MiSeq, which is Illumina platform, and then the other one is nanopore. For both of those, we're using a standardized Arctic primers that basically, kind of amplify certain or sequence certain things and all that. And by using bioinformatics, we basically stitch them together and then run it through, put it through a certain degree of boot camping or bootstrapping, so that we have a higher degree of confidence in terms of our reads and stuff like that. But we are doing these two different parallel systems to make sure that we get a high accurate consensus in terms of our base calls.
We have a few more questions in the chat box. I'm afraid we are having a session.
Venugopal sir is available, sir.
Is it?
Venugopal sir, please.
Not clear.
I think there is one question like in the chat box. And this is from Dr.
Venugopal sir, are you able to hear me?
Hi.
Not clear.
Thank you, sir, for joining. Thank you, sir, for joining. Sir, can you please put in Sir, can you?
I do see our close Dr. Venugopalam. I think he has a problem with his audio, but I got his question here.
Yeah.
And Dr. Venugopal, really nice to see you after a while. We haven't talked after after COVID. But you know this very well. You have visited us here, and collaboration is something that we actively seek. Not because just we want to collaborate, but because collaboration kind of gives you a perspective sometimes you oftentimes not see. There might be some blind spots, and with collaboration and new ideas infused into the whole thing, I think we can be much more effective. I mentioned about our possible work on population-based collaboration between the Spanish group and us. Especially, we want to look into the rural areas of Nepal and rural areas of Spain and how SARS-2 affected these two different communities, and why. And so that kind of question is, I think, is very, very important because we know very well that we live in a very globalized world, and what happens here is going to move very, very fast everywhere else around the globe in a very short amount of time. And so with that, I think, Professor Venugopal, we would want to work a lot more closely with you and your group with COVID-19 also, but other stuff that we've been talking as well.